Shri Gurubhyo Namaha. Welcome to our online learning initiative, and I welcome you all for the biology class. Last class we saw about back cross and test cross, and now we are going to see the other crosses. Before going into the crosses, we'll see about the laws which was proposed by Mendel. So these laws, we call it as principle of inheritance or laws of inheritance. So totally three laws are there. First law, law of dominance, and second law, law of segregation. Third law, law of independent assortment. Okay, we are classifying it as three laws. And out of these three laws, the worldwide accepted law is nothing but a law of segregation. Why they have to accept that law of segregation? Because it states the purity of gametes. So we'll see what is it. So the universally accepted law of inheritance, you can see, it is without any exception. Everybody accepted this law. Why? It states that each trait consists of two alleles. Okay? It consists of two alleles which segregate during the formation of gametes, and one allele from each parent combines. So I'll draw here. It is nothing but, for example, male is having a character with the tallness, and this is the allele for tallness, genotype, we know already. And this, how it is segregating? It is segregating as one allele, another allele in each chromosomes, and this you call it as segregation. And this states the law of segregation, and it states the purity of gametes. What is that purity of gametes? Here it is from the male, the same way another thing from female. So only one will get transferred from the parent to the offspring. Okay, so one gamete from here, another one from there, from the mother's side, right? So next, law of segregation. Okay, just now I said the law of purity of gametes. So because a gamete carry only a recessive or a dominant allele, but not both. You can see here I've taken capital T, capital T. So you may not find any difference. Both are dominant. The same if I'm going to take two different. Okay, a heterozygous condition. Capital T and small t. So capital T in one gamete and then small t in another gamete. That's what they are saying here. Because a gamete carry only a recessive or dominant allele, but not the both. You can see here, right? Not the both. Only one it will be carried. Next, what is law of dominance? The law of dominance says only the dominant character will be seen in the F1 generation. What about the other character? Here, if you see, I'll just show here capital T and small t. It is a heterozygous condition. Both the characters are there, capital T and small t. But in the F1, first generation offspring, it will have only the dominant character, even though if it is having the recessive character. Suppose this parent, okay, when they intercrossed, the F1 generation, okay, F1 generation happen to have capital T and small t. Okay, what will be the phenotype of this particular character? It will be tall only. Okay, there is no difference because this capital T masks the character of small t, will not allow it to express. Only the tallness will be seen in the F1 generation. So the law of dominance says the offspring always exhibit a dominant trait from the two alleles received from the parents. Okay, the only dominant allele is expressed. Okay, once again we'll make it very clear. Capital T, small t, okay, and then small t, small t. So two parents will take this as a male and this as a female. When it is intercrossed, we'll pull out the gamete, okay, to show law of segregation. Capital T, small t, okay, and then small t, small t. Why we are pulling? We are pulling out the gametes so that only one gamete get transferred, right? So we'll take here. So these are the gametes, and from here, one from the male, another one from the female. Okay, the gametes intercrossed, it's transferred, and we'll get capital T, small t. Okay. Now what character it is? The character you call it as tallness only, even though it is having a small t, a recessive character. Okay. So that's what, once again, I will read. It will be very clear for you now. The offspring always exhibit a dominant trait. So this is the offspring. Offspring we are denoting as F1 generation. From the two alleles received from the parent. So what are the two alleles? The allele capital T from male and small t from the female, two parent. Okay. The only dominant allele is expressed. So here what is the dominant allele? The allele which exhibits the character of tallness. Only that is expressed here. So it is tall, right? So let me erase this so that you can see this clearly. Yeah. Characters are controlled by discrete units. So what is that statement? Law of dominance, still more in detail, point wise I've given. Characters are controlled by the discrete unit. It is nothing but gene or factor according to Mendel. Factors occur in pair. Again, I'm saying factor, the term was given by Mendel. It's nothing but gene only. Okay, who coined the term gene? We know already it is Johansen, right? And this factor always occur in pair. So again, okay, we'll take the well-known example, the two factors capital T, and it is occurring in pair. Then, in a dissimilar pair of factors, one member, okay, dissimilar pair of factors, only one member of the pair is dominant, while the other is recessive. For that, just guess. I'll give you 1 second time. Meanwhile, I'll write here. Check what it is. So it is a dissimilar. Capital T, small t is a dissimilar, otherwise called as heterozygous condition. So in that, what is expressed? Tallness is expressed. Now, the second law. Second law states what? It is about the segregation, purity of gametes, how the gametes get transferred, okay, how it is being divided from the parent, okay, and then how this gametes getting transferred from the parent to the offsprings. So we'll see here the two copies of each chromosomes will be separated from each other. So I'll write here two copies. Again, the same example I'm going to write. Capital T is a male character, we'll take it, okay, tallness, and the two copies of each chromosome capital T will be separated from each other, causing the two distinct allele. Okay, you can see here distinct allele separately, right? So here we'll move here, capital T, okay. This is nothing but the law of segregation, right? Located on the chromosome to segregate from one another so that it will be easy to get transferred to the next generation. Now, these two laws, okay, are well verified in monohybrid cross. So totally two cross we are going to see. One is monohybrid, another one dihybrid. By the term mono, it's nothing but single. Hybrid, a combination. So you're taking one trait. When the term comes mono, it is nothing but one. Okay, only one trait, one contrasting character you're going to take, and that contrasting character hybrid variety you're going to get it, right? This is called monohybrid. So I will write here, clearly check, and please make a note of it. So from here, I'll start from here. Parents, okay, parents I will denote with their symbol male character, another one female character. So in male character, again I'm going to take homozygous dominant, capital T, it is nothing but tall character, okay, and then female I'm going to take as dwarf character, okay, and it is nothing but small t. Now, according to law of segregation, we are going to segregate the gametes. So here, capital T, and then here, small t. Okay, and now, so here you will write gametes. You can write just one letter or you can write the full gametes. And now, one from here, this parent, another one from this parent, and it is nothing but capital T, small t. Okay, I'm just showing like this. And then same way here, one from this, another one from here. So it is capital T and small t. So what you will get, 100 percentage. What character you are getting? Tallness you are getting. Okay, 100% tallness. But if you see the genotype, it is heterozygous condition. You're getting capital T as well as small t. Okay, so here you will write F1. This is F1 generation. Okay, F2 generation, you can use a checkerboard. Already in back rows, I told about the Punnett square. Okay, who simplified this and gave a checkerboard for easy calculation. So we'll follow the same. In this monohybrid, use other color. Okay. So since it is both capital T, small t in both the case, male as well as female, I can write wherever I wish. Male, another one female, with the character capital T, small t. Similarly here capital T, small t. Okay, once again I'll go back to the slide. You just check it out. Parent, gametes, and then F1. Okay, we are completed first generation F1. And this F1 we are intercrossing. Okay, you can put a symbol here, intercross. That is on self. We call this on self. We are intercrossing these two, F1 with F1. So I'll write here, note down this term also, on self. Okay. So this character, capital T, small t, I've taken there. Okay. So we'll go to that slide. And now here I'm going to complete as I did for back cross and test cross. Okay. Capital T, okay. Then capital T, small t. Then capital T, small t. Here when it comes, be careful because both are recessive character, small t, small t. Okay, once again we'll check whether we have written correctly. So capital T, right? And then capital T, small t. Then capital T, small t, small t, small t. Okay, all these things, what character? We'll write the character also here. Okay. So the phenotype. We know the genotype, now the phenotype. Capital T, tall. And then capital T, small t also tall. Then capital T, small t also tall because they have dominant character capital T. And here capital T, small t, dwarf. Okay. Now two ratios we are going to write. One is phenotypic ratio, another one genotype. So I will write here phenotype and I'll write here genotype. Just observe phenotypic ratio. Okay, phenotype. What is phenotype actually? The physical appearance. So physical appearance, you can see only two things. Two parental character only we are getting. We are not getting any new character. Tall, dwarf. So how many tall here? Just will count 1, 2, 3. So 3 tall. And how many dwarf? 1 dwarf. So this is the phenotypic ratio. It's an important 2-mark or 3-mark question. Okay, the way of question will be in a different way. I'll train you. Don't worry. And then next, genotype. When you take here genotype, only their genetic makeup. Okay, their genotype ratio or genotypic ratio you can say. Okay, it is nothing but we are going to see this genotypes, capital T, okay, that let me write here. So capital T is 2, capital T, small t is 2, and small t, So how many are there in the number? We'll count. So how many capital T? Only 1. How many capital T, small t? You can see I will encircle it. Okay. So we have 2. So we'll write the number 2 here. Okay, and how many small t? 1. So what is the genotypic ratio? 1:2:1. Based on these ratios, phenotypic and genotypic of monohybrid ratio, we are going to move further. And at the end of the cross, dihybrid cross, I'll tell you a trick for easy calculation of this phenotype and genotype. When it comes to trihybrid or tetrahybrid, whatever it is, In a just simple way, you can calculate it. So we'll move to the next cross, dihybrid cross. Dihybrid, what I said, di is nothing but 2. Mono, 1. So you're taking one character there, intercrossing, and you're getting the offsprings. And here, two characters you're going to take it, right? Di, two characters we are going to take it. So you listen here, the law which obeys this dihybrid cross is nothing but independent assortment. You know the meaning for segregation. We saw the segregation of the gametes there. Dominance, okay, the dominant character is seen in the F1 generation. So, what is this independent assortment? Independent. They are not going to depend on each other. So, I'll give you an example. Just check here. With this example, we'll read the statement. Okay, like the same thing. Chromosomes will draw. Okay. And in this, the character, one is stating for the tallness, another one for color of the cotyledon, yellow color, capital Y, capital Y. Okay. Now we will read the statement and we'll make it still more clear. The traits inherited through one gene, okay, will be inherited independently of the trait inherited through another gene. Okay. So, with this statement I'm explaining here. So, one gene, okay, when it is coming and expressing, okay, it will say for tallness only. Another gene it is coming and expressing, it will say for the color of the cotyledon, yellow color. So, they are not dependent on each other. They are independent. Okay, the traits of one gene is independent of the trait of another gene. Again, I'm making it still more clear. The trait of tallness gene is not dependent on the trait of color, yellow color gene, right? So they both show their character independently. And this you call it as a law of independent assortment. Okay? And why they are given the term meiosis? That cell division takes place in by this process only, meiosis. Okay, it is nothing but reductional division. So we'll see a dihybrid cross here. Okay, the same way for monohybrid we have done, right? The same way with the parent, gamete. But here, what is the twist? Okay, what is the change is nothing but two things we are going to take, two characters. So I'm going to take color of the cotyledon, yellow color. Okay, shape of the cotyledon, round. So I'll write the phenotype here, yellow, round. Whenever you are writing the character, write both phenotype and genotype. And here, small y, small y, small r. Because in the beginning I told you what letter you are taking as a dominant, the same you will take it in a lowercase as recessive character. Okay, irrespective of the color or variation. So I'll write here, it is nothing but what color? It is green. And the recessive is nothing but wrinkled. Wrinkled. Okay, green and wrinkled. So here I'm going to pull out the gametes. Okay, so I'll write here the letter G. Why R? When you're writing the gamete, two gametes you have to take. The gamete of the color, the gamete of the shape. The gamete of the color is yellow color and the gamete of the shape is round shape. So both you have to write. Don't do mistake in the exam. Sometimes what you will do in a hurry, you will write YY itself, okay, and then RR. Okay, it's not so. So write both the characters like this. Okay, once again I'm writing here YR and circle the gametes. Same way here, small y, small r, small y, small r. Okay, now your F1 generation, you are going to self the F1 after this. First, we'll complete the F1 generation. So you can see here, why while you're writing the four, okay, the characters, You have to pair it. YYRR. Don't write YR. Okay, that is wrong. So YYRR. You may ask, Miss, the same thing you can do. YR. Why it is wrong? Because when it is like this, it is not showing their character. YR, what character it is? It's not a character. It should be in a pair. The alleles should be in a pair. Yellow means YY. Round means capital R, capital R. Okay, it should express like this. This is wrong. You are not supposed to do like this. Okay, the same way here. We'll do it. Okay, capital Y, small y, capital R, small r. And try to find out what mistake I have done in this. Okay, you can check one mistake. Okay, I've written here both the same thing. Okay, what you have to do, the character, one character you're getting from the parent, male parent, another character you're getting from the female parent. So where is that small y and small r? This is yet another mistake you'll do in the exam. Okay, as I'm doing and correcting it here, you will get it in your mind. So correct it. I'll erase this. Okay, so be careful while writing. So capital Y, small y, capital R, small r. Got it? And now this we are going to self. On self. Okay, on intercrossing with this, how it is. So there only one character, which was very easy to do that Punnett square. And the gamete also only one gamete. But here two gametes you're going to take. When it is all dominant, all recessive, it was so simple. YR. Either capital Y, capital R, or small y, small r. When it comes like this in heterozygous condition, okay, you have to be still more careful and pull out the gametes. So be with me and listen. Capital Y, it duplicates, okay, like this. Twice, okay, that's what I said the word, duplicates. And then the small y, again, duplicates. Okay, like this small y. Write it. And then now this R, okay, will join. I'll use another color so it will be still more clear for you. Yes. Capital R and the same capital R. Okay, you will not write for the same capital Y. Again and again, you will not join with the same dominant character. This will go for the recessive here. Okay, capital R. So capital R also duplicated, and how it has joined? One with the dominant, another one with the recessive. The same way, small r, the recessive character, you duplicated. One here, okay, with the dominant, okay. Another one here. Yeah, another one here, small r. So you can see four gametes. 1, 2, 3, and 4. Got it? 4 different gametes. See the combination. All the 4 duplicates, but how they are combining? One dominant, one recessive. One dominant, one recessive. Or both dominants, both recessive. Like that. Okay? So you can see here the 4. We'll move to the checkerboard now. So here the same way. Okay? I'll show you the previous slide. Just check. I've done for 1. Okay? Another one you will do it and complete it. The same thing only you will get it. Right? And this gamete, one for the male, another one for the female. So totally 4 gametes for the male, another 4 gametes for the female. We are going to do in a checkerboard. So we'll write here male, okay, denote with the symbol. Another one female. So we'll write capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. One from the male gamete. Another one from female gamete. You'll write here. Capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. Children, I will do one, okay? And you're going to complete rest of the thing and you will get the ratio, okay? So one, we will start here. Capital Y, capital Y, capital R, capital R. I told you already, when you're writing it in the four, okay, you have to write the pair with pair, right? Capital Y, capital Y, capital R, small r, same way, okay? Then capital Y, small y, capital R, small r. And the rest you are going to do, okay? By the time I'll also complete here. Capital Y, capital R. Be careful while writing the letter. You may go wrong if one letter is wrong also. Small r, y, small y, capital R, small r, capital Y, small y, small r, small r. Same way you find here capital Y, small y, capital R, capital R. After doing once, you have to check where anywhere whether you went wrong. Capital Y, capital R, small r. So here small y, capital R, capital R, small y, capital R, small r. So capital Y, capital R, small r, capital Y, small r, small r, small y, capital R, small r, small y, small r, small r. Okay, a random check I'll do. Okay. Yeah. Here, this one is a recessive, pure recessive. From here, we'll see this is nothing but small y, green. I'll write just the starting letter green. What it is? Wrinkled. Okay, green wrinkled. And then here, if you see the combination, small y, is there. So it is green, round, 2 capital R. So it is green, round. We have one dominant here. Green, round. Okay, so we got a new character. We have taken green wrinkle, but here new character green round we are getting. Okay, similarly, any other new character, let us check here. So here, yellow wrinkled. We have taken yellow round. Why it is yellow wrinkled? I'll write only the abbreviated thing. Why it is wrinkled? Because small r. Anywhere like that. So here, yellow wrinkled. Anywhere like that? Here, yellow wrinkled. Remaining all, if you check, okay, everything will have either one dominant character or all the dominant character, or in out of this, see, I'll show you one thing. See, one dominant, one recessive, like heterozygous condition you're getting. So all these things will be yellow round. Okay. So the phenotypic ratio. Okay, you can figure out the phenotypic ratio here. Okay, you will get 9 is to 9 is to 3 is to 3 is to 1. Okay. Next class, we'll see about genotypic ratio. Okay. Here phenotypic ratio, how many? 9 yellow round, and then 3 green round. Okay. And then 3 yellow wrinkled, 1 green wrinkled, you got it. Okay. Next class we'll see about the tricks also, how to calculate this phenotype and genotype. Thank you.